Hello, my name is Ana Maria Agudelo and today I'm going to present the Seminario of Molecular Biology. The article is Effect of the Protein Corona on the Transfection Efficiency of Lipid Code-Graphene Oxide-Based Cell Transfection Reagents. First of all, to introduce the article, we are going to talk about the transfection. The transfection is the process to deliver a nucleic acid into an eukaryotic cell, and we can do it by these three ways. The first one, most of the cells are treated with calcium phosphate or DEAE dextran that form a complex with the DNA, promotes their addition to the cell surface. The second one is the liposuction, and the DNA are treated with cationic lipids, and this complex can diffuse in the membrane. And the third one is electroporation. The DNA are incubed in special vials that contains electrodes, and then they apply a current, and this current makes the molecule more permeable. The second thing we are going to talk about in the article is the protein corona. The protein corona is the result of the interaction between the GOCL DNA that are called grapholipoplexes and biological fluids such as human plasma. Then the complex exposed to a protein get involved by a layer that are called protein corona. And the layer involved with protein concentration leading to biocoronator with modified cytochemical properties. The objective of the article is to explore the effect of the protein corona on the transfection efficiency of lipid code-graphene cells transfection reagents. One of the methods that are used in the article is the preparation of GOCL nanoparticles, and the fundamentals are the nano-assembler TM benchtop that employs a microfluidic cartridge and pumps two different solutions into two microfluidic channels that converge in a terminal channel by fine-tuning mixing parameters as total flow rate and flow rate ratio. And the purpose of the method is to obtain an homogeneous solution of graphene oxide and cationic lipid nanoparticles. The other method is the cell culture, and the fundamentals are that the GAMB, the MCF7, and the U87 cells were preserved in the Lenko's modified Eagle medium supplement with 10% of fetal bovine serum, and the CaCO2 cells were preserved in the Roswell Park Memorial Institute supplement with 20% of FBS. And the purpose is to freeze and preserve the cancer cell lines, the glioblastoma cell line, and the colorectal adenocarcinoma cell line. The other method is the transfection efficiency experiments, and the fundamentals are treat the cell lines MDAMB, MCF7, the U87, and the CaCO2 with the GOCL DNA complex. And the purpose is to carry out the transfection to analyze the luminerase expression and determine the transfection efficiency. And the last method is 1D SDS phage cell electrophoresis. And the fundamentals are that the 1D SDS phage is a method that separates protein by molecular weight. In this method, samples are weighed and dissolved in sodium dosed sulfate, that is SDS. SDS is a negative charge detergent that has both hydrophilic and hydrophobic regions, and the proteins are completely denatured by the SDS. And the purpose of the method is the protein corona characterization. So this is one of the results of the article, and this is the 1D-SDS phage gel electrophoresis that shows the comparison between the GOCL nanoparticles and the grapholipoplexes after the dispose of a different amount of human plasma of 1%, 2.5, 5, 10, 20, and 50% amount of the human plasma. And the most important result is that the protein corona composition of both systems was influenced by protein concentration. And in addition, here we have the M is like the molecular weight that is compared with both particles. About the last picture, in the electrophoresis, they found that a low protein concentration in human plasma, lower than 5%, the protein patterns of GLCL nanoparticles and the grapholipoplexes were almost superimposable. And on the other hand, at high protein concentration in human plasma, lower than 20%, marked differences in the protein profiles of GLCL nanoparticles and the grapholipoplexes were found. And the result about the electrophoresis is that in low protein concentration, they preferentially bind to the lipid surface on the grapholipoplexes. So this is the last result in the article, and we have the 5A, B, and C is the complex GOCL DNA without an amount of human plasma. And in the D, E, and F, we have the complex GOCL DNA with an amount of human plasma of 20%. And the MD and B cells were treated with fluorescently labeled, that is the green, pristine and biocoronated grapholipoplexes. And cells were stained with lysotracker, that is the deep red, that is a lysosomal marker, and the DAPI. The DAPI is the unstain of the nucleus. So, while pristine grapholipoplexes escapes lysosomal degradation, the biocoronated counterpart largely accumulates within lysosomes, and it has been demonstrated the formation of the protein corona provides a capacity to alter both cellular and intracellular location of the nanoparticles. And grapholipoplexes colocalizing with lysosomes gave rise to yellow clusters, like the yellow clusters we observe right here. For the discussion, I take three different authors, and the first one say the evolution provides of size and zeta potential of biocoronated grapholipoplexes report in figure 2. Closely resemble those previously reported for cationic liposomes protein complexes. It's true, is what they say in the article. The second one, in this view, protein corona formed in human plasma may compromise endosomal escape of biocoronated grapholipoplexes should link them to lysosomal compartments. Is true. And the last one, this inversion in surface charge is likely to be responsible for reduced electrostatic interaction with cancer cells that in turn may lead to lower cellular uptake, and that is true. Finally, to conclude, the layer of protein corona formed by a high concentration of proteins can reduce the transcription efficiency in the cancer line cells using the experiment, and also can increase the probability to digest of the DNA useful blood and the intracellular level. And in conclusion, if we expose grapholipoplexes complex to an artificial corona formed by a low concentration of proteins, can increase the transcription efficiency and low cytotoxicity. So this is a concept map about the transcription, and the link is below, so that's it.